Okay. Okay, welcome everybody. So I want to start out with just a little primer on some of the physics, but also the molecules that are relevant in this session today. So we'll be talking about the glycocalyx, and that'll be, I'm not going to delve much into that, but you'll certainly hear all about it during the talks today. And then and the role that it plays in interaction with both leukocytes, interactions with cells, as well as interactions with chemokines. And you'll hear all about that in the talks today. So wanted to start by talking about the people that will be speaking. Professor Handel from UCSD will be talking for basically the first hour, and then Shenda Baker will talk for 20 minutes, Megan Lord for 20 minutes, Megan Priestley, and William Seeley. So you'll see the word heparin a lot in here because heparin or heparin sulfate, heparan sulfate is a major component of the glycocalyx. And so you'll see that, or the words glycosaminoglycan or GAGs. So glycosaminoglycans are sugars, and they form the cell surface sugar layer that is often referred to as the glycocalyx. So there's a lot of words here. I just wanted to show that there are papers out there that describe this interaction of the glycocalyx, or in particular heparin sulfate proteoglycans, as they interact with lots of different molecules. So what we'll describe as the glycocalyx or the GAGs, these molecules are highly anionic. And so my interest has been very much electrostatics related. And so this was a paper about the role of this interactome in cancer. And it talks about all these growth factors that interact with the heparin sulfate proteoglycans. But they also emphasize that these electrostatic interactions with the heparin sulfate recruit chemokines and cytokines that can influence the immune response to a tumor. The same thing goes for the cell surface on not just cancer cells, but in the endothelial glycocalyx, or on the surface of any cells that have this sugar coating. You'll have these electrostatic interactions going on. And so what are the molecules? We've talked about the heparin sulfate proteoglycans, but there's also mucosa. So the mucosa are, mucus is secreted, but it's also tethered mucins. So there are mucins, and there are also these glycosaminoglycans. There's molecules that are highly anionic in biology. These are generally macromolecules. They can be up to, 100 kilodaltons. The big molecules, it can be, or megadaltons in some cases. So they're really large molecules, and they interact with cations then. So if there are a lot of negative charges, what neutralizes that? If you zoom in and you look at the negative charges, there's inevitably some positive charge around. What charge might that be? Well, we know that it might be individual monoions or monovalent cations or divalent cations. And this is a diagram showing the pairing that might happen between, or sort of the preferred pairing that might happen between cations and then these charged groups on macromolecules in biology. So you have carboxylates, you have phosphates, sulfates, and sulfonates in biology, and those are going to pair in a weak type of hydrogen bonding, ionic bonding. So it's not covalent by any means, but this ion pairing these ions, as they approach each other, will be attracted. And in that process, they'll drive out water. So especially if these charged groups on the macromolecules are on, if they're on macromolecules, then as they're being neutralized, you're going to squeeze out some water. So there's a kind of a dehydration event that happens as these are electrostatically attracted to each other. These are diagrams showing as you move these ions together, the distance between them. This is the free energy between them at different distances between them. So what this shows is a lysine and a lysine and a glutamate, and here's an arginine and a glutamate, and you can see that the energy is below zero, meaning it's preferred interactions with a contact ion pairing or a solvent separated ion pairing. And you see that with arginine in particular, there's this bidentate pairing that means that they really like to be attracted. And the solvent separated has an energy well, but then if it squeezes out that water, you end up with this deeper well that's just contact ion pairing of the ions. So you might see this sort of contact ion pairing between the various negatively charged groups, and the arginine or lysine that occur. So now it's not just sodium and potassium that will neutralize our glycocalyx, but it's also these charged groups. It might be on peptides or other molecules, as you'll hear about. So wanted to mention briefly that, our plenary speaker today is one of her colleagues at UCSD had worked and published with others in 2020, this idea that SARS-CoV-2, one of the things that it binds to is a heparin sulfate molecules. So here comes in that virus, and when the virus comes in, it first encounters the heparin sulfate proteoglycans on the surface of your cells, and then it will find the ACE2 receptor, and there's other dynamics that go on. But the binding to the cell surface is a really important feature, and you can see why. If you look at SARS-CoV-1 versus SARS-CoV-2, the positive charges are in blue, and so it becomes increasingly cationic. And there's another group that early on found that everybody heard about this furin cleavage site, and the furin cleavage site was cationic. And then when Omicron came by, it picked up 9 extra positive charges on the RBD region of the spike protein. And so it keeps getting more and more cationic, which means that it's going to bind to the glycocalyx of cells better. So the evolution of the SARS-CoV-2 showed that it got bluer and bluer here. And so one of the things I wanted to show today, just as an intro, was that chemokines have these cationic, conserved cationic amino acids, which you'll see here as R's and K's. And this is a bunch of the neutrophil activating chemokines. And you'll see R's and K's that are conserved across all of them, or many of them, and there's slight differences. This was a study that was done where they mutated the arginines. And when they mutated the arginines, the tightness of the tightness of the binding of the chemokine loosened up as they mutated. So they sort of found that it was naturally compact with these glutamates interacting with lysines and arginines. And all these dotted lines are ion pairing events between glutamate and lysine or arginine, which was that other slide I showed earlier, showed how as they approach each other, they find a distance of contact ion pairing. So with that, so keep in mind then the ion pairing that happens with these large, primarily hydrophilic macromolecules, the electrostatic interactions. As they occur, they're driving out water, and evolution will work to make the binding optimal for whatever function is going on. So with that, wanted to introduce our first speaker. Let's see, I think I wanted to mention, I think that's 10 minutes, 11 minutes, so I should get going here. So first speaker is, I actually want to stop this, start and stop recordings at every juncture. So for each speaker will have a their recording. So I'm going to stop that.